Now we will see the conversion of inosine monophosphate to adenosine monophosphate and guanosine monophosphate. Say this is ribose phosphate, ribose 5-phosphate. Okay. This is inosine monophosphate, IMP, inosine monophosphate. From this inosine monophosphate, aspartate, using aspartate and GTP, GDP. N ribose phosphate NHCH CH2 COH COH. Okay. This is such an aspartate attaches to the by removing carbonyl oxygen of the carbonyl carbon. Okay. Now this is adenyl succinate. Adenyl succinate. Adenyl succinate. Then in the next step, adenyl succinate or adenyl succinase. Adenyl succinase enzyme converts it into adenosine monophosphate by removing fumarate. Ribose 5-phosphate. Rest all fumarate is eliminated, so that we will get adenosine monophosphate or AMP. Okay. So inosine monophosphate by using aspartate and GTP, it is converted into adenyl succinate. Adenyl succinate immediately by removing a molecule of fumarate by adenyl succinase enzyme converts into adenosine monophosphate. This is AMP. Then similarly IMP, inosine monophosphate dehydrogenase. Inosine monophosphate dehydrogenase enzyme converts inosine monophosphate to xanthosine monophosphate. Here NAD, point say NAD is reduced to NADP. Okay. Then ribose 5-phosphate, this is xanthosine monophosphate. Xanthosine monophosphate. Xanthosine monophosphate. And next enzyme GMP synthase or synthetase, GMP synthetase enzyme by utilizing glutamine and ATP is required, glutamate plus ADP. Second position, carbonyl oxygen is removed by replaced by NH2 group. Ribose 5-phosphate. This is guanosine monophosphate. So this is a conversion of inosine monophosphate to adenosine monophosphate and guanosine monophosphate. Inosine monophosphate by utilizing aspartate and GTP, it is converted into adenine succinate. Adenine succinate by removing fumarate by adenyl succinase enzyme, it is converted into adenosine monophosphate. Similarly, inosine monophosphate dehydrogenase converts inosine monophosphate to xanthosine monophosphate. Here coenzyme NAD is reduced to NADP. The reducing equivalents are accepted by NAD. Then xanthosine monophosphate by utilizing a molecule of glutamine and ATP and by the enzyme guanosine monophosphate synthetase, it is converted into guanosine monophosphate. Glutamine is converted into glutamate. ATP is converted into ADP. This is the conversion of inosine monophosphate to adenosine monophosphate and guanosine monophosphate. Here we have to remember one thing. PRPP glutamyl amidotransferase is an enzyme which is controlled by a feedback mechanism. The feedback mechanism, it is controlled by or inhibited by purine nucleotide itself. Okay, PRPP glutamyl amidotransferase enzyme is controlled by feedback mechanism by purine nucleotides. Okay. Then coming to the salvage pathway. Salvage pathway means phosphoribosylation of pre-existing nitrogen base. So adenine, phosphoadenine is there, NH2, N. See, this is adenine. Adenine, enzyme adenine phosphoribosyl transferase. Adenine phosphoribosyl transferase. Phosphoribosyl transferase transfers as a ribose 5-phosphate to adenine forming AMP. Ribose 5-phosphate. So salvage pathway means phosphoribosylation of pre-existing nitrogen base. So thereby adenine, by utilizing adenine phosphoribosyl transferase enzyme, a ribosyl 5-phosphate is attached to the nitrogen at 9th position, so that we will get adenosine monophosphate. Similarly, for guanine, enzyme hypoxanthine guanine phosphoribosyl transferase. Hypoxanthine guanine phosphoribosyl transferase. Thereby we will get GMP, guanosine monophosphate. Okay, guanosine monophosphate, GMP. See, this is the salvage pathway. So one more thing is there, xanthine, hypoxanthine. hypoxanthine, hypoxanthine guanine ribosyl transferase. Hypoxanthine guanine ribosyl transferase enzyme transfers ribosyl 5-phosphate to hypoxanthine, forming ribose 5-phosphate, forming inosine monophosphate. These are the salvage pathway. See, salvage pathway means phosphoribosylation of pre-existing nitrogen base. To adenine, phosphoribosylation occurs. Enzyme adenine phosphoribosyl transferase transfers a ribose 5-phosphate to 9th nitrogen, so that it will form adenine monophosphate. Similarly, for guanine, enzyme hypoxanthine guanine phosphoribosyl transferase enzyme transfers a ribosyl 5-phosphate to 9th nitrogen, so that it will get GMP. Similarly, ribose 5-phosphate is transferred to hypoxanthine to get inosine monophosphate. Okay, this is the salvage pathway of purine nucleotides. Okay, this is all about the biosynthesis of purine nucleotides. Thank you.